Hi, welcome to our Immunology Workshop Group presentation. Here we consist of 7 members, which are Dylan, me myself, Azrin, Felicia, Ma Hongwei, Chenchi, Karina, and Nur Putri. For this case scenario, Kathy is a 35-year-old woman just confirmed that she is pregnant. Instead of feeling happy, Kathy is feeling worried because she had 2 miscarriages last year without any known causes. No additional tests were conducted to investigate the possible reasons of this miscarriage though. It is desired to visit fertility clinic for counseling so they can be more prepared on what they can do to ensure successful pregnancy this time. Note both Kathy and her husbands are healthy adults living a healthy lifestyle. This is our hypothesis. First is hormone imbalance. Second, blood type, RH incompatibility, probably in placenta. Third, genetic factors. Four, environment factors. Fifth, urinary abnormalities. And lastly, age-based factors. For our hypothesis, we decided that Kathy might have RH incompatibility with the fetus. This condition occurs when RH negative mother is pregnant with a fetus who is RH positive. Although the fetus RH blood group was still unknown, the fetus may have inherited the RH positive from the father.
The genotype. The ABO blood group system is determined by the presence of A and B antigens on the red blood cells. The Rh factor refers to the presence or absence of Rh antigen, which is also known as the D antigen, with Rh positive being the dominant over Rh negative. Blood group genotypes includes A, IAIA or IAIO, B, IBIB or IBIO, AB, which is IAIB, and O, which is IOIO. The Rh factor genotypes are Rh positive or Rh negative. Kitty's blood group is B, IBIO, and Rh negative with anti-A and anti-D antibodies in her serum. Her husband's blood group is A, IAIO, and Rh positive with an anti-B antibody in his serum.
So I will be discussing the concept of phenotype. So a phenotype is an observable attribute of an organism such as blood type, influenced by both genotype and environmental factors. For instance, let's look at Kathy and husband's blood types. Kathy's blood type is B negative, whereas her husband's blood type is A positive. These blood types determine the presence of specific antibodies in their blood. Kathy's blood contains anti-A and anti-D antibodies, while her husband's blood contains anti-B antibodies. So to understand their blood types and antibodies, it is very crucial, especially given Kathy's history of miscarriages and her current pregnancy status. This knowledge can help us to manage any potential complications, ensuring a safer pregnancy journey.
Hi, now we'll be talking about the hemotypes and blood group. First, let's understand the ABO blood group system. There are 4 main blood types, A, B, AB, and O. Each type is defined by the presence or absence of specific antigens on the red blood cell and antibodies in the plasma. So, for the type A, it has A antigen on the RBCs and antibody B antibodies in the plasma. Type B has a B antigens on the RBC and antibody A in the plasma. Type AB has both A and B antigens on the RBC and no anti-A and anti-B antibodies in the plasma. This makes AB the universal recipient, meaning they can receive blood from any ABO type. Now let's talk about O. O has no A or B antigens on the RBC and both anti-A and anti-B antibody in the plasma. This makes O the universal donor, meaning they can donate blood to any ABO type. Let's look at the case study to understand this better. Kathy has a B negative blood group, meaning she has anti-A and anti-D antibodies in her blood serum. Her husband has a A positive blood group, meaning he has anti-B antibodies but lack anti-D antibodies. When a husband and wife have different blood types, it can lead to complications during pregnancy. ABO incompatibility is really less severe and can cause mild hemolytic disease of a newborn or HDN. However, RH incompatibility can be more serious. If an RH negative mother, like Kathy, carries an RH positive fetus, it can lead to severe HDN. This is why it's crucial to understand and manage these blood group incompatibilities during pregnancy.
Let's look at the consequences of RH incompatibility. Firstly, the impact on pregnancy. Hemolytic disease of the fetus and newborn, HDFN, may occur if the maternal antibodies are able to cross the placenta. These antibodies then will destroy the red blood cells of the fetus. The impact on maternal health is that pregnancy complications may also arise, including severe fetal anemia and hydrops. These conditions require close monitoring and often leads to early delivery. What about the impact on fetus and newborn? Firstly, it can lead to fetal anemia, as due to maternal antibodies is passed into the fetal's bloodstream, leading to the red blood cells to swell and rupture. This causes the decreased oxygen delivery to fetal tissues. Next, hydrops fetalis is characterized by severe swelling in the fetus due to severe anemia. Excess bilirubin, known as hyperbilirubinemia, can also deposit in the brain, leading to kernicterus. Furthermore, organ enlargement may also occur as the liver and spleen need to grow to compensate for the destruction of red blood cells. Lastly is heart failure. It is the result from the fetus' effort to compensate for severe anemia and hydrops.
Treatments for RH incompatibility are divided into two parts, which are the treatments for the mother and the fetus. Firstly, the RH negative mother can be managed by using RH immunoglobulin RHIG therapy called ROGAP. This prevents sensitization of the RH negative mother to RH positive fetal blood cells by stopping the mother's RH negative blood from making antibodies that attack RH positive blood cells. The mechanism behind this treatment is that HIG contains antibodies against RH antigen, clearing fetal RH positive cells from the maternal bloodstream to prevent the formation of antibodies and administered at around 28 weeks of gestation after potential sensitizing events and within 72 hours postpartum if the baby is RH positive. Next, the corticosteroid therapy is given to accelerate fetal lung maturity in case of anticipated preterm delivery due to severe RH incompatibility and is usually administered in the late preterm period at around 34 to 36 weeks gestation. If delivery is imminent, to reduce the risk of neonatal respiratory distress syndrome (RDS). And as for the fetus, the treatments for the fetus are intrauterine transfusion treatment for severe fetal anemia due to RH incompatibility, where transfusion of RH negative blood cells directly into the fetus to counteract the effect of RH antibodies when severe fetal anemia is detected via ultrasound or other diagnostic tests. Lastly, phototherapy is the treatment for neonatal jaundice if bilirubin levels are elevated postnatally by keeping the baby near fluorescent lights to reduce bilirubin buildup in the blood.
During pregnancy, Katie carrying RH-positive fetus, blood from fetus enter maternal circulation, this causing a process called alloimmunization that the maternal immune system sensitizes the foreign RBC antigen from fetus. Next, maternal immune system then starts to produce IgG antibodies against the foreign antigen. But this amount of antibodies aren't enough to affect the first pregnancy, but are prepared for the future exposure. In subsequent pregnancy, with another RH-positive fetus, Kathy's immune system quickly recognized the RHD antigen and produced large amount of anti-D antibodies. These IgG antibodies cross the placenta and enter the fetal bloodstream. Maternal IgG antibodies bind to the RH-positive antigen on the surface of fetal RBCs, causing the destruction of the fetal RBCs and leading to hemolysis. Severe anemia reduces the oxygen carrying capacity of the fetal blood, leading to hypoxia and death. On the other hand, the destruction of RBCs release large amount of bilirubin, which can cross the blood-brain barrier, causing damage and potentially leading to death.
RHIG should be administered to negative pregnant women to prevent the formation of anti-D antibodies. This is particularly important at 28 weeks of pregnancy to ensure that if infant blood mixes with maternal blood, RH-LG is already present to avoid sensitization. In case where the baby is Rh positive, RH-LG should be administered to the mother within 72 hours after birth to prevent his immune system from attacking Rh positive cells in future pregnancy. For every potential synthesis event, include miscarriage and abortion, Rh-negative mothers should receive RHLG to ensure their future pregnancies are protected. Early in pregnancy, both parents should have their blood type and Rh factor tested. Rh-negative mothers should have regular antibody screening to detect any anti-D antibodies.
This is our reference. That is all from us. Thank you.